Welcome back. I'm Professor Cong. Let's study the development of modern pathogen biology. With the development of many new technologies, such as biochemistry, molecular biology, cell biology, bioinformatics, etc., pathogen biology has also been rapidly developed. The history of pathogen biology in the 20th century is merely the discovery of genetics, nucleic acid, biochemistry, and molecular biology. So there are 4 aspects that I want to introduce to you the development. First is the genome sequencing promotes genomics research. The second is the development of pathogenic biological diagnosis technology. The third is about drug and vaccine development. The fourth is modern organisms as research models of life phenomena. The first is the genome sequencing promotes genomics research. The first complete genome sequencing, known as phage genome sequencing, was completed in 1977. Haemophilus influenza's genome was sequenced in 1995. The first draft of the human genome was then obtained in 2001. So far, more than 200 bacteria have been sequenced, such as Haemophilus influenza and Mycobacterial tuberculosis and Helicobacter pylori. The genome sequence of C. elegans and the study of the genomics and proteomics have driven the development of the whole life science to a great extent. At present, the parasite genome sequence has been completed, including Plasmodium filaria, Trypanomyces, Schistosoma japonica, Leishmania, Toxoplasma gondii, Cryptosporidum, micro Anopheles, gubbin, and other parasites, laying an important foundation for the research of parasite functional genomes. And the second is the development of pathogenic biological diagnosis technology. In recent years, some fast, specific microbiology diagnosis methods have established, including recombinant DNA technology, enzyme-linked immunosorbent assay, and polymerase chain reaction, and the development of monoclonal antibody technology brought about a revolutionary change in clinical microbiology and allows the use of the training method to diagnose, not cultivate the organism. Gene sequencing of pathogenic organisms can not only better understand their structure and function and pathogenesis, but also provide basis for the screen of effective clinical anti-infective drugs and vaccines. Third, let's see the drug and vaccine development. Due to pathogenic biological resistance makes it necessary to drug research. Development of vaccine is to prevent infections, disease, epidemic, and minimum the harmfulness of the powerful weapon of the current vaccine. The current vaccine type merely has weak poison vaccine antigens, secreted vaccine, genetic engineering vaccine, and synthetic vaccine and DNA vaccines. There are 30 more kinds of human vaccines have been used so far. Fourth, the model organism as research models of life phenomena. So, some organisms have become favored objects of many scientists as model organisms. Examples such as Cephalopods elegans and fruit flies are highly successful model organisms that have helped several scientists win Nobel Prize. So let's see the great discovery in pathogenic biology. In the process of the development of pathogenic biology and related disciplines, there are nearly 60 scientists won the Nobel Prize for outstanding contributions. Let's see how the great scientists discovered their great discovery. The Julius Bonder. His discovery of factors in blood serum that discovery bacteria. This work was vital to the diagnosis and treatment of many dangerous continuous diseases. So he won 1990 Nobel Prize. So Charles Nichol, his work on typhus, he found the vector of typhus is the body rules. So he won 1928 Nobel Prize. So let's see Thomas Morgan. He was called father of fruit flies because he got the great discovery in the role of chromosomes in heredity, and the theory of chromosome of heredity was established in Drosophila, the fruit flies. So let's see these three scientists, Sidney Brunner, John Thomson, and Robert Harvard. They used Cephalopods elegans to uncover the mechanisms of gene recognition in organ development and programmed cell death, to discover the genetic regulation mechanism of organ development and programmed cell death. So they won 2002 Nobel Prize. So Chinese scholars also made a significant contribution to this pose. In the 1930s, member of Chinese Academy of Scientists Aaron Huang, in the study of encephalitis virus from animals of the virus in the process of cultivation, improved to the in vitro tissue culture cells, which led to a foundation for modern virology. In 1955, Shang Fen, everyone who uses the chicken embryo yolk sac vaccination and the streptobacteriosis method, isolated from the world's first virus strain, known as two virus Chinese scientists. Chinese scientist Tu Youyou discovered artemisinin and has made outstanding contribution for malaria treatment. In a landmark in the history of human malaria, won the 2015 Nobel Prize in Physiology or Medicine. Become Chinese scientist Nobel Prize Zero Breakthrough. So for these lectures, we study the prevalence of infectious disease, the discovery of pathogen of biology, and the classification of pathogenic organisms, and the harm of pathogenic organisms to humans. And we also know the development of modern pathogen biology. And we also know great scientists that made great discovery to these regions. So from these lectures, we know the importance of this class and the infections and immunity is so important for the disease. Okay, let's see you next class. Thank you.